All right, guys, we're going to try something a little cool here. I'm a little excited. Um, we're going to go through a lot. Let me go up here. We're going to talk about eukaryotic cells now and go through all their organelles. But I don't want to do it in that just like boring way of just going through a list. I kind of want to make this into a little story, which will hopefully like reveal to you like how crazy all of this is. And you wouldn't get this with any other professor at the college or probably many other schools. So it's going to take a little while, but I hope you'll get some extra juice out of it, and it'll land with you a little more, and you'll just see how crazy this stuff is, how absolutely bonkers we are. Okay, so we got a little prokaryote here. This is a little E. coli bacteria. It's got its little flagellum. It's got its nucleoid. It's jam-packed full of protein. And this is essentially what I'm showing you here. So this little guy right here, these little dots here, they're a little prokaryote. That big cell, these are red blood cells actually, right here. And this is a white blood cell. It's a neutrophil. And you can see how smart it is. It can actually smell the bacteria and it can follow it. You see it turns there and is chasing this bacteria to swallow it up. It's pretty nifty. Pretty nifty indeed. And you can see it's about 20 times bigger than the bacteria is. Bacteria about 2 microns. Neutrophil is about 20 microns, something like that, maybe a little less. Pretty wild. Okay, so if a bacteria is in the bloodstream, a neutrophil can just find it. A white blood cell can just find it. But let's say the bacteria is outside of the bloodstream, like it's right underneath the skin. Well, then the neutrophil has a bit of a problem. And we're going to walk through cell biology and the function of a cell based on this problem of how does the neutrophil, so this would be the white blood cell, this is the white blood cell, this would be the wall of the vein, let's say, actually be a capillary, we can just say the vascular tissue. So these are the cells of capillary or vascular tissue. So these make up the kind of thin parts of your cardiovascular system. That's what these would be. And then this is obviously a little bacteria. How does the white blood cell know how to when to leave the bloodstream? How does the white blood cell know when to leave the bloodstream and search for bacteria to swallow? All right, so I've not taught this like this before. This might be kind of bananas. I think it's gonna be cool. But we're gonna walk through this, and then I have a video to kind of show you at the end, and hopefully that video will then make sense. So this white blood cell, we're gonna kind of just start out here at the surface. You know, it's kind of rolling along, it's kind of crawling along. These endothelial cells, that's what these are called. These are called endothelial cells. Cells that make up the walls of your vascular tissue, of your veins, your arteries, and the thin parts in between called capillaries. All right, and then where there's bacteria, these endothelial cells have begun to express some special surface proteins that stick up like this. And these will bind to surface proteins within the white blood cell. So now this white blood cell knows, hey, oh, boom. These surface proteins communicate to the white blood cell, there is bacteria on the other side. Okay, lecture's over. Problem's done. Right? That was the question. How does the white blood cell know when to leave the bloodstream in search of bacteria? These surface proteins communicate to the white blood cell there's bacteria on the other side. All right, we're done. But what happens now? That's what we're going to walk through. Okay? So these will create a signal. And this signal now goes all the way to the nucleus. We're not going to draw out all of that signal, but there would be, let's say there's proteins, just for simplicity, let's say there's proteins associated with these guys. And now they leave, doop, and they go to the nucleus. So once we have binding occur here, these little proteins leave and they go to the nucleus. All right. So I want a thin arrow. So binding of surface proteins sends a signal to the nucleus. Pretty nifty how that works. That's how a lot of cell signaling works, actually. So a lot of times these signals just go directly to the nucleus, either through a little protein. Sometimes there's molecules like steroids, which they will just go right through that membrane and then go right through that membrane and signal into the nucleus. Okay, so now we're going to leave the surface and we're going to go into the nucleus. All right. Well, the nucleus, I've kind of drawn it incorrectly here. The nucleus actually is made out of two membranes, an inner and an outer membrane, like so. So the nucleus is actually made out of two membranes. And I know this might be kind of hard for you guys to draw if you're not doing it, if you're doing it in a notebook. You know, feel free to have this kind of like overhead view here. And then you can, everything that we're zooming in on, you could just draw that, you know, on another page. Be like, zoom in of the nucleus. Okay. From the nucleus, we're going to go to the ER. Now we're at the ER, new page. Okay. So feel free to just flop pages and write that this is a zoom in or a view of. Okay. What is stored in the nucleus? We have DNA. DNA is stored in the nucleus. And let me see if I can pull up this little picture. That I had originally. It's actually not very good. I'll pull up a different one in a minute. Okay, so inside the nucleus we have DNA. We have it in two different kind of forms. We'll draw the DNA in blue. And we have two different forms of DNA. We've got DNA that's tightly wrapped around protein. So we'll make the protein actually. We talked about this a little bit with the rats. And then we've got DNA that's loosely wrapped around protein. All right, and then we'll draw the DNA in blue. I'm not going to draw the strands here just because it's kind of like a zoomed out view. So this wraps around, wraps around, wraps around, wraps around. That one kind of trash. Let me fix that. It actually wraps around twice, but I'm just kind of doing this quickly. So we got DNA wrapped around protein, and then we got DNA loosely wrapped around protein. Struggling to draw with my mouse here. Okay? And the whole nucleus is going to be full of protein. We have the whole nucleus is full of protein. Nucleus is like, let's say like, nucleus is, I don't know, maybe like 2 to 10 micrometers in size. and it holds approximately 2 meters of DNA. So think about that. It's pretty crazy. If you don't know what a micrometer is, it's very, very small. It's 1,000 times smaller than a millimeter. It's crazy. So it's 1,000,000 times smaller than that, and it holds that. And the reason why it can do that is because it can wrap this DNA around these proteins to condense it, to organize it. So this over here is called heterochromatin, and this is called euchromatin. This is tightly packed. This is loosely packed. This loosely packed, genes are on. They're able to be on. Tightly packed, genes are off. Okay? So this is one way that we can regulate gene expression. Pretty cool. All right. A couple other little nifty things about the nucleus. One is that the nucleus has, its membranes don't actually look like this. Well, I've already talked about these, but they actually loop back on each other. Let me erase that there. So the membranes are actually the same size. I drew them different sizes here just because I did. But they actually look like this. So they actually loop back on each other. Whoops. Might be a little hard to understand because this one's thick and this one's thin, but it will look something like this. So they loop back on each other. Let me see if I can actually find a picture of that just so you can see what I'm talking about here a little more clearly. Yeah, and then we're going to draw that guy in next. Okay, this is coming up here. There we go. All right, so you can see this. This is the nuclear membrane. One layer, 2 layers, and they loop back on each other like that. 1 layer, 2 layers, and they loop back on each other like that. And then we got this crazy thing in there called a nuclear pore. And this is like a big freaking basket. You know, and it's embedded right inside of this membrane. I'm going to draw it kind of in like 2 sections. Like so. Like these little sections wrap all the way around, of course. I'm kind of leaving that center part open. And they actually have like little feelers that come out of the top too. Got little feelers that come out of the top. And then down at the bottom, inside, it kind of makes this like loop and this little hoop looking thing, like so. And then we would have another kind of, you know, version of this going around the back like that. This thing is a nuclear pore. Nuclear pore. Which allows things in and out of the nucleus. And these are all over the nucleus. Okay. Got another little image here I want to bring in. So here we've got our nucleus. And you can see all these nuclear pores all over it. So we got these nuclear pores embedded in this double membrane that kind of loops back. If we look directly at a nucleus with an electron micrograph, you can see all of these pores within the nucleus. And they actually look like this. They look like little flowers. And they can decorate the entire nucleus. There's so many pores. Okay? All right, so now our signal is going to come in likely through a nuclear pore, if it's a protein signal. So our signal is going to come in through this nuclear pore. I'm not going to redraw the whole thing, but I'll redraw it quickly up here. So we got our nuclear pore. Looks something like this, like that. So our signal protein can come in. Our signal protein from the surface enters the nucleus. Frickin' cool. Okay. And now what is it going to do? Let's say it actually binds to DNA and takes the gene and it turns a gene that is off into a gene that is on. Pretty freaking cool, right? So now we have a gene that is on. All right? And genes that are on can be what's called transcribed. So let me make this a little smaller here. So we got our gene here. This would be called transcription, this process, taking DNA, converting it into RNA. RNA, if you remember, is also a nucleic acid. It is single-stranded. Single-stranded. And it acts as a temporary messenger. You've probably heard of messenger RNA. So that would be mRNA, messenger RNA. So we're going to make an mRNA here. Okay, and this actually will then be converted into a different kind of protein. All right. I think that's all I want to talk about for the nucleus. There's a lot more I could talk about, but we take this part of the, let's say, the DNA. This is obviously DNA, maybe not obvious to you, and we're converting it into RNA. There's a whole nother protein and machinery that does that. It's called DNA polymerase, but you don't need to know about that. Not right now, anyway. Okay, so now we made an RNA. We got our single-stranded RNA of our gene. This is our messenger RNA right here. Because let's just kind of review what we did here. Boom. These endothelial cells touched proteins on the white blood cell. And those things said, hey, we got bacteria out here. Proteins associated with those proteins left. They went into the nucleus through a nuclear pore. They turned a gene from being off to being on. And then that allowed RNA to be made. Okay, cool. We're done with the nucleus. That's the nucleus. All right. Well, actually, kind of we're not done with the nucleus. Psych. We kind of are and we kind of aren't. Okay, so kind of crazy thing is that associated with the nucleus is another thing. So we're going to leave that. We're going to come back to it. Let's draw this next little guy in. So actually attached to the nucleus, you could really actually think of it as an extension of the nucleus. It's not really actually separated. But we're going to draw it separated because we do think of it as being separated. is another structure. And it has a lot of folds. And I'm going to draw it really just kind of very simple and just kind of small like this. That seems pretty good for now. We'll draw the other side. Pretty cool looking. I don't hate it. All right, and then we will continue off on this side, let's say. Actually, we'll just leave it at that for now, okay? And then attached to this thing is all kinds of little dots. Like so. It's very membrane-y. This is just a membrane, it's very folded. Very cool. Okay. This thing is called the endoplasmic reticulum. It's kind of a crazy name. I have some song rolling through my head about the endoplasmic reticulum, but I don't know what the beat is to. I'm sorry, it doesn't matter. Okay. Well, okay, we'll actually get to it here in a second. Okay, so the endoplasmic reticulum, short for that, we can just call it the ER, because that's a mouthful. It's a cool science word. And this one is actually the rough endoplasmic reticulum, because it has these little dots on it. And these little dots are something called ribosomes. Ribosomes, they read mRNA and translate it to protein. They are the site of protein synthesis. You will have to remember this in this course. They are the site of protein synthesis. Pretty crazy. They themselves are made out of protein. And as a little side note, there's kind of a, you can actually see it right here. This little kind of dense part in the nucleus is called a nucleosome. This is the site of ribosome production. I know, it's a lot. Too much, too many words, too many things going on. All right, just settle down. We're doing okay. It's all going to make sense. Okay, so now what happens? Well, this RNA, we want to make, kind of weird how it does this, but we want to make a protein out of this. So what does this RNA do? Well, it's going to slide out of this nuclear pore. So the RNA, boom. Messenger RNA, mRNA, leaves nucleus through nuclear pore. And then it has two options. Depending on the RNA, there are ribosomes floating around out here. I will be right back, actually. Okay, I am back. We've got our RNA that leaves the nucleus, messenger RNA. And now it has two options. One, this RNA can be translated by a ribosome in the cytoplasm. That's this fluid out here in the cytoplasm. So there are ribosomes out here just floating around too. So like this is a ribosome, boom, boom, boom. So it could be translated by one of these and turned into a protein on here. But since this protein is eventually actually going to go, that we're going to make, it's actually going to go back out here to the membrane and then out to the outside of the cell as well. There's going to be a couple of different proteins that are made. So this proteins that are going to be put on the surface of the cell or outside of the cell are always made in the endoplasmic reticulum. So let's write that down. Proteins that go to the surface or outside of the cell are made in the ER, specifically the rough ER. So that means that this messenger RNA is going to be read at one of these ribosomes here. So we're going to have our surface RNA right here, or our messenger RNA right here, and it's going to go literally through this ribosome like a piece of computer tape or ticker tape, and it's going to be read by it. And then it's going to be spit out, and then it could actually go through another ribosome and be read again until it is degraded. And so as it's being read, what happens is this ribosome then starts to add on the different amino acids that are being read by each piece of the DNA code that is now coded in the RNA. And remember that RNAs are going to fold up, or sorry, proteins are going to fold up. And we're going to get this three-dimensional shape of this protein from these various amino acids. And let's say it looks something like that. Okay? So that's our protein. So now our protein is being synthesized by a ribosome at the rough endoplasmic reticulum, the rough ER. Okay, and this will happen a couple times, many times. It's constantly happening over and over and over, actually. And that is how we are making our proteins that are going to signal for this white blood cell to kind of change its shape and exit to go fight this bacteria. Okay, so we get enough of these little green proteins. Let's say we get a bunch of them start to form right around here. What happens is part of this endoplasmic reticulum is actually going to start to bud off. So I'm going to erase this here, like so. And we're going to redraw in this gray. But part of this endoplasmic reticulum is going to start to bud off, and we're going to have all of our little green proteins that are kind of folded up, kind of in here like this. Pretty nifty. And there'll be some other proteins that come with this too. Maybe we'll draw those in red. That are just like, you know, they're not on the membrane, they're just kind of in there. All right. Then, eventually this buds off. This whole thing buds off. And now we've got a little specialized structure. Boom. Like so. And we'd have all of our green guys in there still. Oops. Draw these more simply, so I don't take forever doing this. And we've also got our little red guys folded up in here, let's say, from a different messenger RNA. Okay, so now we've got a vesicle. A vesicle buds off of the rough endoplasmic reticulum. full of the freshly made proteins. Pretty cool, right? So, what did we just learn right here? Rough ER is the site, one of the sites, is one site of protein synthesis. Another site, I don't have time to, God, I could teach a whole class on this. It's sad that I don't have more time. Synthesis. The cytoplasm, the other site of protein synthesis. Okay, those are our two sites. The rough ER and the cytoplasm are where proteins can be synthesized. So for example, we could have messenger RNA being read by that guy, and we could be making a little orange amino acid coming off of this, little peptide coming off of there. Okay, pretty cool. Okay, great. Let's just keep cruising. Let's just keep on going. It's going to be a long one, but I'd rather just keep cruising through it, and you guys can pause it as you need to. All right, another little key. So this stuff can continue on. ER can continue on, and it does. So the cell will not just have rough ER, it'll also have more ER, but it doesn't have ribosomes. So this is called smooth endoplasmic, smooth endoplasmic reticulum. And this is the site of lipid synthesis. Also, like some detoxification and things like that occur in the smooth endoplasmic reticulum. We're budding off lipids here. We need to make more membrane, let's say out here. We need to be constantly synthesizing new phospholipids and things like that. So we'll make new phospholipids here, just for example. So we also have a smooth endoplasmic reticulum. That's all I'm gonna talk about there. It does not contain ribosomes. All right, very cool. All right, so now what do we got? Now we're moving on. Now we got the Golgi stacked up. Golgi stack, pancake up, stack protein. That's what's going on at the Golgi. All right, so now this little guy, this little vesicle goes on to something called a Golgi. And the Golgi apparatus looks like a stack of pancakes. Bellyache, bro. All right, so we got one little stack. We got another little stack. Golgi stack, pancake up, pack protein. We got another little stack. And another little stack. Ooh, that looks kind of hard to parse with my brain. It's so green. Okay, so this guy comes on over here. And this vesicle then fuses with the Golgi. Okay, so then like these proteins and all that stuff go in here. So we'll have these green proteins. You know, they'll be in here like this. They get fused in there. And then we got these guys also in there. And what happens is little vesicles butt off again. And then they go to this stack. And then the vesicle goes to that stack. And the vesicle goes to that stack. So vesicle will fuse with the Golgi. Vesicle will fuse with the Golgi. And then contents, so then the proteins are sorted, modified, and packaged for delivery. It's pretty nifty. It's like the Amazon warehouse of the cell. And this is the Golgi apparatus. Golgi apparatus. That's what that's called. It's got a cool name. So as it goes through the stacks, the proteins get more modified. And like modified, what does that mean? I mean, maybe they get a little sugar added to them, let's say. Maybe they get a couple little sugars added to them or something like that. So we know that these are self. So something that can happen is carbohydrates can be added to them. All right, it's just one little example. They can be cut, they can be clipped, they can be folded different, all different kinds of things. And then eventually what we get is we get a vesicle coming off with our modified proteins. I'm just going to re-copy this one and paste it over here. We get then the final packaged, properly made proteins budding off of the Golgi apparatus. The Amazon warehouse of the cell. You know, it's like Amazon, FedEx, everything all together. They also do some modification. So it's kind of like the production factory too. This is more like the factory. The rough endoplasmic reticulum is more like the factory. And this is like the modification, sorting, shipping center. Okay, bang. We're cruising along here, actually. All right, now this vesicle is going to go all the way, it's going to be transported all the way over to here. And how does it do that? These vesicles aren't just floating. They're actually walking on things. Big ass. Sorry, didn't mean to cuss. They're walking on huge parts of the cytoskeleton called microtubules. And we'll just bring it down here, actually. Make it a little easier for me. Okay? And these little microtubules, we'll talk about these. Are like little roads. So we got a microtubule. Microtubule. It's like the little highway system. Of a cell. Okay, but instead of having cars walk on these little highways, there's little frickin' people that walk on these. So we've got a tiny little person called a kinesin motor. So they kind of have a little structure like that, and then they got a big old foot like this. And then we got another one. Oops. It's got a big old foot, something like that. And this boy literally walks this vesicle to the surface. Pretty freaking cool. So this kinesin motor is going to walk this vesicle all the way down this microtubule. It's just not just one kinesin motor, a bunch of kinesin motors will do this actually. All right. All the way down here. And then when it gets down here, this vesicle is then going to fuse with the membrane. Boom. Boom. Kinesin motor walks vesicle packed with protein to the cell membrane for delivery. It's like a little delivery boy wearing a backpack. If you've ever played some Death Stranding, it's like Norman Reedus from The Walking Dead carrying a bunch of crap on his back and falling over. It's really funny. Okay, so now this vesicle, this is actually pretty cool. This vesicle will then, it's got some stuff on the outside. I don't have time to go into all this, but it's got some stuff on the outside. These little guys will actually intertwine with each other. They're called snares. There's snares here. And they will wrap like this around it, twist like rubber bands to bring these together until we have a fusion of these two membranes. So now these two membranes will actually fuse together. And I'm just going to draw it over here just so you can see what's going on. Let me erase this membrane down here. And then we've got this gray membrane with the vesicle contents like that. And then we've got our little green proteins. Like so. And these will now, I'll draw one more little version of this. And then we've got our red proteins starting to get dumped out just into the extracellular kind of matrix out there. And I'll draw one more little version of this over here just so we can see what the fusion looks like when it's complete. It's kind of cumbersome to erase. Like so. And this will all be gray now. That's where the vesicle membrane added on. And then we've got our little green proteins now expressed out on the surface, like so. And we've got our red proteins dumped out, like that. Okay? It's pretty crazy what we've just done. We've just had these proteins bind, we've gone all the way to the nucleus, we've made new RNA, then we've made new proteins in the ER, we've packaged and sorted it in the endoplasmic reticulum, and then we've shipped those proteins out to the surface so that they can then do their job. These proteins would then bind, these would be like an integrin actually, and they would bind to, they would have to be closer here. I kind of drew these in a bad spot, but these would bind to their own, like a little collagen fiber or something like that. And then that would signal to the protein to start to change, signal to the cell to change its shape, to crawl through the blood vessel. It wants to now go through it. All right. So that's what the cell would now do. How would it change its shape? That's a pretty interesting idea. It's pretty crazy. trying to think if I want to stop this or if I want to keep going. I think I'll keep going. I think I'll keep going. Okay, so that's the basics there. Let me actually stop this one. We'll watch this little video. Yeah, let's watch this video and then I'll come back and I'll talk about the cytoskeleton. Okay, so this video is actually, we're gonna watch a video that kind of encompasses everything that we just saw. So, all right, I don't know where I paused it. We're gonna watch this video though from the top. Let me hit play. We're gonna watch a little animation on everything we just kind of went through so far. So this is called the inner life of the cell. This is the white blood cell here. We're in a blood vessel. These are red blood cells. This is the surface of the white blood cell. These are those surface proteins interacting with each other. And this kind of starts the whole cascade. It says, oh, you should come through here, white blood cell, because we got some bacteria. We're looking at a bunch of different stuff here, some signaling molecules, all this jam. That's a little lipid raft. These are little spectrin filaments. You can see those spectrin cages there, which control diffusion within the membrane. This is the actin cytoskeleton, which is just below the surface of the membrane, helps give the cell its shape. We're going to talk all about that. This is an actin filament polymerizing, becoming a polymer of actin monomers. Again, so we got an actin filament being cleaved here now by another protein. Got a microtubule, another cytoskeleton. This is the road that the kinesin motors walk down, and they can depolymerize, as you see there. Got our kinesin motor walking on a microtubule, carrying a vesicle. Yeah, they walk like that. That's pretty cool. It's pretty cool indeed. Got a lot going on here. We got the big nucleus back here. That's the centrosome. That's a mitochondria. So those are the nuclear pores, and RNA is coming out of the nucleus into the cytoplasm to be read by a ribosome. So we've got a ribosome locking on. Boom. Making a protein, RNA, ribosome protein. Let's pause that. RNA, ribosome, protein. And the ribosome is a protein too. All right, we got those new proteins being synthesized, doing their thing. That's a mitochondria back there. Now we got a ribosome docking at the endoplasmic reticulum. Not the smooth, but the rough endoplasmic reticulum. Some of us like it rough, you know what I'm saying? So they hang out on the outside and they squirt the newly synthesized protein down in to the ER. You can see that happening right there. Boom. And then it goes and falls apart. Now we got vesicles budding up off of the ER. They're going to be shipped to the Golgi. We got our little kinesin motor again. So now we got our Golgi coming in here. We got our vesicles coming in on our microtubules to fuse with the Golgi. We got our vesicles coming off, walking down microtubules to leave the Golgi. This is our Amazon shipment center. Got our vesicle. That was kind of fast, a fast transition. So we got our vesicle getting walked all the way out to the membrane now. Microtubule, kinesin motor, vesicle, and it's going to fuse it with the membrane and dump the contents out on the surface. So some of those proteins go out into the extracellular matrix, and some of them stay in the membrane, right? And they're fused to the membrane, and they're going to bind to stuff. They actually accumulate with a different lipid. It's called a lipid raft. They're slightly taller lipids. And then these are going to bind to these collagen fibers out here and these other proteins. And that's going to say, hey, cell, change your shape, get flat, and crawl in between these endothelial cells. So now it gets flat and it crawls in between these cells. It's pretty cool. It's pretty cool indeed. All right, we just learned what the heck all that was. Very slowly we drew all that out. So this process, this cell will now crawl. The white blood cell will now change its shape and crawl between the endothelial cells. It has a very special name. It's called diapedesis. Diapedesis. Dia means across. This word means across. And pedesis, this word means walking. So the neutrophil will walk across the endothelium. Means walking across. All right, and we'll come back in another video here and talk about the cytoskeleton, which is important for changing the shape of all of these, of the cell, so that it can now walk across. We'll go through the 3 different cytoskeletal elements and describe them in their details. Shouldn't take more than 20 minutes. I'm not gonna lie. It's gonna take a little time. Sorry this is long, but you know, we got a lot to cover. I mean, look at that. Look at how cool that is, man. We just did all that. That's pretty wild. Inner life of the cell. You can now understand the basics of this video and what it's actually trying to communicate. Isn't that cool? I think that's pretty cool. All right, I'll see you in the next one.